All right, welcome back. And I know we got people wandering in from lunch, so we'll let them wander in. But we're going to go ahead and kind of get started because we got a lot to do in an hour. So we have printed out copies now of this document. I have one here, and I put one at each microscope station upstairs. So we have 3 microscope stations upstairs. And what we're going to do is set up, in a moment, we're going to set up the slide preparation right back there. The first thing you need to know how to do is actually set up your microscope for use. Okay, so just out of curiosity, how many of you have used a microscope at least once before? Okay, good, very good. Yeah, so what it says here is after the first page, it has all your equipment and so forth, and it has a thing on using, storing, and transporting your microscope. Make certain you read that. If you have to transport, especially, one of the biggest things to know is that you don't want your objective banging into your stage. So what we do is we rotate, even if you're moving it around here, rotate for your smallest objective, like your 4x. So if you look at it here, I currently have the 40x objective down. If I were going to move this, I would turn it like this, so the 4x objective is down, and then I would lower the stage so that they can't accidentally bang into each other. That's important. You can mess up a objective that way or scratch it up and so forth. So there's a lot of other stuff about packing and storing and so forth, right? Then there's cleaning slides and slip covers before and after use. Very important. See what we're seeing right there? That's a 40x, because I've got the 4x right now. It's a 40x magnification. What do you see?
Dirt.
That's a dirty slip cover and a dirty slide, right?
That's dust.
Yeah, it's dust and dirt and everything. And if we go to there, give it a second. That is 600x.
Oh, the dust is mostly gone.
Yeah, now we're looking at really small pieces of dust. Okay, what you're actually looking at there, that's the edge of the slip cover.
Oh, okay.
Yeah, and so what I'm using this as just a way to make certain that I can get everything really crisp. And because the way I've got this set up right now, the challenge is that the focus in here is just a tiny bit off from the focus up here, but I can just run it like this with no problem. All right, so what you're going to see is that we are going to clean our slip covers and we're going to clean our slides before we do anything else with them. What we're going to do is we're going to have, over at the slide prep station, we're going to have your clean water and a pipette, the clean pipette, and then you'll use the clean water. We probably also need a little dish or something that like when we rinse the slip covers and the slides, that water can just go down into.
Use distilled or just regular water?
Ideally, we would use distilled water. What we're doing here is that Tactical got us some spring water and ran it through a 0.5 micron or 0.1 micron filter. So it should be nice and clean, okay? But we're going to clean off and make certain that we get it. But that particular pipette and that clean water, it never touches anything else. All it's used is to add clean water into this, okay, and to rinse other things. Never pick up anything with it or whatever, otherwise you're going to get nasties in here that will get into your samples, okay? So we got one pipette that's going to be used for that. The other pipette is what we call your sample pipette, and it will go into the other water, and it'll be called the rinse water, okay? And so that when we rinse it out after we've taken a sample. And then these graduated test tubes, they have milliliter graduations on them, and that's what's going to let us see how much put in, okay? Okay, slide used to make certain they're free from scratch, scarred, any damaged slides or slip covers if the damage would interfere with proper use. Slide and slip cover are not completely cleaned before use. Rinse them well with water. from the clean water pipette. Dry them gently using a lint-free cotton cloth or one of these optical wipes, right? A sample slide can dry out after it sits in the microscope for a little while, resulting in the slip cover becoming somewhat stuck down to the slide. To clean the slide, simply irrigate it gently with water from a clean water pipette until the slip cover floats free. You can then separate the slip cover from the slide and wash each with clean water individually. Never try to pry the slip cover up when it gets stuck. You'll just break it. The most fragile thing in this whole deal are the slip covers, okay? By far. The slides are actually pretty robust. And these are the slides we'll be using right here, okay? And they're pretty robust and hard to lose and so forth. What I will do is I won't set them down here. If I need to set them down, I'll set them down on the microscope's stage so that I know where it is. It doesn't get mixed in with things on the table, okay? Same thing, if I have to temporarily stow a slipcover, don't put it down because you'll never find it again. They disappear.
You only hold it around the edge?
You only hold it on the edges. So you notice I'm only holding this on the edge, and when we'll clean it, and then everything we do, we'll hold them on the edges. And the same thing is true of the slipcover, okay? Now, the slip covers that we're using here are 18 by 18 millimeter, and that's very specific. So we're gonna play with that. And if I take and pull this slide off and push this slide on, okay, hold on. Give us a slightly thicker slide. Okay. There we go. Okay. You'll notice that all of a sudden, if I roll around, okay, I'm not seeing all that nasty. Looks pretty clean, okay? That's the difference between a dirty slide and a, this one we've had out, we had some other stuff in it, and it's just got a bunch of gunk on it. So I just pulled that one out of my case just to kind of show you the difference, okay? Glass slip covers are especially delicate, must be handled with care. Glassware that breaks creates nasty cuts. This stuff is really it's deceptive. It will so if you be very careful with them, and if one of them shatters, be really careful with the shards. They're just super sharp, okay? And try not to drop it on the floor because they're hard to find on the floor, and they will end up cracking and having little sharp pieces of glass all over the floor, which is not good, okay? So out of everything we're doing, be the most careful with the slip covers because they're the things that will give you the most trouble, okay? Now, preparing the microscope for use. We're all going to have to do this when we break up, and we're going to choose a station and then go to the station, and we're going to try to get them. We do need one power cord for one of the microscopes up there. It's just a standard 3-prong power cord. I don't know if we can find an extra power cord for that. We'll find
a TV cord.
I brought
Yeah.
a bunch of them. Oh, like a TV?
Yeah.
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 Yep. We need one of
Somebody
those.
punch buttons and I'll go get one.
we can get them in a few minutes. I'm just going to need one at some point.
Okay.
Okay. So every time you use the microscope, use the following setup steps. Remove the dust cover. Now, what does that step imply?
That you put it on.
That you put that you put the dust cover on when you finished the last time. Yes. Plug in the illuminator and turn it on. So the illuminator, most of the new ones have LED illuminators. Okay. You plug them in, you turn them on, and there's usually a dial on the side to change the amount of illumination. Okay. Rotate the 10x or 20x objective into position. Place a sample slide on the stage and focus until the sample is clear. In the ideal world, I didn't bring my sample slides with me, but usually what we'll do is we'll have some prepared sample slides, and then as I'm doing some just immediate sanity checking, I'll just pull out a sample slide, throw it on here, and check it out to make certain everything is still looking good, right? So you can buy a prepared sample slide for a couple of bucks from these different scientific houses. And they can be great for teaching children too, okay? So You can buy a whole little set and tray for a few bucks. But it's really good to do that. And it helps you just make certain that everything is working right. Then for microscopes with focusable condensers, you can now focus the condenser by placing a piece of paper in contact with the light source so that half of the light source is covered. Move the condenser up to the maximum height and then slowly move it down until the paper edge comes into focus, and then remove the paper. Who's done this step before? Okay, this is one of those little optimizations. If you'll notice, if I take this and put it right over that, let's see if I can get it in here and keep it steady. Well, so I'm going to back up to a, we'll back up. Let's go back here. Okay.
Oh, yeah.
Okay. See that? Now, if I get it down in there and hold on to it, it's actually easier to see. What's happening is it's the auto white balance is becoming problematic because I've taken half the illumination away from it, but I can see it really clearly here through the eyepiece. And what I can do is there's actually a over here, there's a little dial for the condenser, which is this little thing that focuses the illumination. And you can bring it up and down. And if it's not in focus, if it's way out of focus, you want to move the condenser up and down a little bit until you get that. Because what that's going to do is it's going to make certain that all the illumination from the illuminator is nice and focused right where you want it on your sample. And that's going to give you nice, even illumination that's well focused and not like messy. Okay? So that's one of the things that I would recommend doing is to take the moment to make certain that your condenser is in focus, right? Now, the next thing to do is for you, adjust the distance between the two eyepieces so that you get a good binocular image when looking with both eyes. So these will spray out and back down. And so this is how you adjust for your own interpupillary distance, right? Everybody know that term? Distance between your pupils. I happen to have fairly a little narrower, so I have to kind of pull mine in pretty far. And then, like if Chase is working with me, he'll have to pull it out farther because where his pupils sit. But you need to. What I'll do is take my dominant eye, and I'll put it on, and then I'll move until I can take my subdominant eye and get it equally matched in. And so, by the way, if you wear glasses, yes, you take them off the same way you do with binoculars. And then that's why it's, for me, a little bit different focus here than here because I'm using this focus to compensate for my eyes, okay? So when I look at it, yep, I want to adjust it until, aha, there we go. I've got a really good stereo image. Now, if it's your microscope, you especially want to take time to your left has this, your right. And the same thing is that these microscopes will usually have, most of the microscopes that aren't super cheapy will have a way to compensate for the diopter. And usually the diopter is on the left eye because so many people are right eye dominant. So what you do is you figure out where the diopter is, and here it is on my left eye. So what I'm going to do is I'm going to close my left eye, I'm going to look at my sample with my right eye, and I'm going to focus until that sample is in laser focus. Then what do you think I'm going to do? Switch eyes. That's right. I'm going to close my right eye and open my left eye, and it might be that that left eye is now out of focus. Right here on this eyepiece is the diopter adjustment. Now adjust the diopter until your left eye is in focus. What we've just done is we've just dialed in your prescription to the microscope so that both eyes can see nicely, okay? That's an important step. Otherwise, you'll be like, why are my eyes going crossed and I can't see anything? It's like, well, because, you know, both eyes needed slightly different focus, okay? Then you should be able to like switch back and forth between eyes and both of them be in focus. That's what you want. And then when both eyes open, it should be really clear stereoscopic image where you're not like looking through half the half the eyepiece like this and half of it's like you should see a really clear, very well focused field. These are the small little details that make the difference between being able to see the details nicely on your slide and be like feel like you're fighting with the microscope and why is this not working, okay? So all these steps are documented for you in the document, right? And then now after that's all set up, you are going to go to taking field samples of soil or compost, right? So we take the sample from 1 to 3 inches below the surface, gather shortly before the intended time of you're going to examine it. Do not let it sit out in the sun or to dry out. Take samples from several locations. It's a good idea so you don't get fooled by something unusual going on at a single sample site. So it is possible that you could go out and you could say, I'm going to sample from right there. And that happens to be the point that last night one of the dogs decided to go and pee. Okay?
Bad luck.
Bad luck. So that would throw our results quite often to so forth. So the standard sampling procedure is to take several samples from different locations, combine them, and then work with that. So what we do is that lets us, yep. You missed diopters, Jack. We talked about diopters.
I don't know what those are.
So we want to sample, and as a matter of fact, when we're doing this for field science, there's actually a sampling protocol to make certain that we eliminate as much of that sample bias as possible. We take multiple samples. So each sample has multiple sample sites, and then we have multiple samples, and we come back and we look at all of them, and so forth. Once you get good at it, it'll probably just run out and get a couple small little samples, mix them together, and come in and take a look at it, and so forth, right? When sampling a compost pile that is actively in process, try to gather a sample from the warmer area at the center of the pile in order to get a good look at what is happening in the area most likely to go anaerobic. A good compost pile should remain fully anaerobic all the way throughout. That's, of course, if you're not doing an anaerobic. I hate to use this term. There's no such thing as anaerobic compost, but it's used colloquially. Composting is an aerobic process and fermentation is an anaerobic process. And so, yeah, it's when we started talking about fermentation yesterday, one of the things that to be pointed out is that, yeah, part of the reason we keep it underwater is so that there's no oxygen, and when you're fermenting, that is a microbial transformation, metabolic transformation of some substance in anaerobic conditions by anaerobic microbes. In soil, we are favoring aerobic, and when we get into an anaerobic condition, we want to go back to aerobic as quick as possible generally. There are some forms of anaerobic composting that are designed to help you break down materials that are impossible to get to stay aerobic over time. Example, I met one of the state resource officers from Alaska. She had gotten a call that a whale had beached right outside of one of the communities. And they were like, what are we going to do with this, right? Because it was already starting to smell a little bit. Okay. Don't blow it up, right? Not a good idea. So the problem is that what are you going to do with that, right? Well, the thing is, it's actually got a huge amount of organic matter in it. And so what her solution to it, which was brilliant, she's like, we're just going to anaerobically, we're going to ferment it basically, but we're going to call it anaerobic compost. She got a front end loader and dug into the side of a hill, and they just pulled the whole whale up in there and buried it and came back 2 years later after it had gone through this anaerobic decomposition cycle and then turned that into an aerobic compost before using it. So when we do an anaerobic compost, what we do is we can use it. And Bokashi is one of the famous methods that was invented was developed in Japan to be able to take and use and even do things with like meat and materials and so forth that were really hard to keep aerobic and have them go through a decomposition fermentation cycle anaerobically and then go into aerobic, okay? So it was a little side note, but what we're wanting to do when we're doing an aerated compost pile is we're not wanting it to go really anaerobic. And the most likely place for it to go anaerobic is the farthest place from the surface. So when we're doing the microscopy, I'll dig in and get a sample from the most likely place to have gone bad, just so I can make certain that, like, that's my worst case place. I want to go look at it, certain it's still good. Okay, so that's when you're sampling your compost pile, it's what you're going to do. Okay, there's also instructions here we're not going to get into today for preparing a compost. Okay, how should I say this? There is one section here for preparing a soil or compost sample slide. That's what we're going to be doing today. There is down here a whole other set of instructions for preparing a compost tea or compost extract sample slide. Don't do that today. That would be if you are doing aerated compost teas or extracts. This just tells you how to prepare a sample slide under those conditions. Okay, we're going to be doing this procedure right here, and here's what it basically is. Now we're going to double this. Okay, it says place 1 gram of the sample in a test tube, add 4 grams of clean water. I figured out with these particular test tubes that we have, there's no mark for 1. So what I typically do is just double it. We do 2 and 8 to come up to 10. Okay. So what we're going to do is we're going to take your sample and we're going to pack your sample down in here until it comes up to the 2 milliliter mark. Okay. Then we're going to take our pipette and we're going to add water until we bring it up to the 10. Then we're going to these tops screw on, and we're going to screw the top on so that when we do the agitation phase, we don't get sprayed with it. Okay. You can technically, in the old days, they used to actually do this. They put their thumb on top like this, and then they would shake. Okay. You sample all over yourself and the lab bench and everything. Today, we like to use these because they screw on nicely. I still hold it like this, right? And it says, okay, it says remove the top from the test tube, use a pipette. Okay, so, step 2. Put the top or stopper on the test tube and shake the tube using a shoulder to waist arcing action for 30 seconds. Where each complete arc cycle takes about 1 second. Okay, and we'll show you what that looks like. No, not this. Okay? Yeah, it's not a shake weight. What we're going to do is we're going to get up here. We'll let Jack figure out what our breathing pattern should be while we do this, right? But you're going to hold it up here, and the arc you're going to go is from here to here. Okay? So this is your arc. And I usually just hold it like this so that it's easy for me to get a hold of. And I'm going to do it this way. I'm typically going to go 1, 1,000, 2, 1,000, 3, 1,000, 4, 1,000, all the way up to 28, 1,000, 29, 1,000, 30, 1,000. Okay? And so what I'm trying to do is without a bunch of scientific equipment, I'm trying to get almost the same agitation every time on a sample so that I'm getting the same amount of agitation of the sample to get the same amount up into the liquid so that if we were doing this in a lab, we would have equipment to do all these steps, right? So it could be really repeatable. And we'd be measuring things with a great deal of precision to be super repeatable and so forth. We're trying to do what we're going to call field expedient microscopy, right? We're trying to look at it and get into the ballpark. If you want to write a lab report on this, then we're a little more careful, so forth. I'm trying to get you good enough that you're going to be able to get qualitatively correct data, okay? So once you've done that, we're going to let it sit upright for a few seconds so that all the large particulates can precipitate out. We want the clay is going to hang in for a while. Don't worry about that. We want the sand and the silt to mostly fall out, which will fall out in the first 15 to 30 seconds. So we just let it sit, and you'll watch any sand, anything else falling out to the bottom. Okay, and what's going to happen is you're going to get this cloudy-looking fluid up here, and you're going to get some solids falling back in there. Okay, so that's going to be your next stage.
How long does this sit for?
Typically, I'll let it set about 30 seconds or so. I'll look at it and see when most of it's kind of come out, right? Okay, then remove the top from the test tube and use a pipette to take a sample from 1 milliliter below the sample surface. Pump the pipette in and out 3 times so the inner surface of the pipette is saturated. Then draw out your final sample. Okay? So what we're going to do is you should have about 10 milliliters of fluid in here by the time we get done with that. We're going to take our sampling pipette and we're going to take it and we're going to put its tip down somewhere between 8 and 9. We're going to pump in and it'll come up. We're going to pump out, pump back in, and we're going to flush it about 3 times. What we're doing is we're making certain that for that particular sample that the inner surface is saturated, and we're not, when we take a sample, we're not pulling away from our sample. Then what we do is we clear it, and then we pull in our real sample on our 4th time. We pull it out like this. Okay, now I should have, a bunch of sample in my pipette. What I want to do then is I want to get the first couple drops out because I want to get anything that got stuck in the lip. I want to get it out of there, right? So what it says is, in this, it says, okay, discard the first drop from the pipette back into the test tube, then place the second drop in the middle of your slide. So we got it pulled out. We're going to go push just enough that we get one good drop off of this, and then hold it just like that. And we're going to go over to our slide, and we're going to put one drop in the center of our slide. Okay? First drop is for the test tube. The second drop is for the slide. Okay? Now we've got a drop on the slide. Now we got to get a slip cover on there. Okay? This is, once you've seen it and done it a couple times, it's actually simpler to do than to explain. But this is why I have the dirty one, so I can demonstrate, okay, and where I can hold them up and you can see them. What we're going to do is we're going to take the slip cover and we're going to hold it just like that. We're going to take and we're going to put the edge of the slip cover right in the edge of the drop. And as soon as you do, what's going to happen is that drop surface tension is going to put across the whole width of the slip cover on that edge you have down. And then you're going to drop the slip cover down onto the slide like that. And of course, it won't go sliding because the fluid's going to hold it in place. And what's going to happen is you're going to watch that fluid basically, the surface tension is going to draw it out across the whole slip cover. Now, the reason we're using the specific size of slip cover is because we want that to be spread out over a known area. We've got one drop, and we want it spread out over a known area, okay? Now we've got a prepared slide. The clock has started. Why is that?
It's going to dry out.
It will dry out, right? What's going to happen is you're going to start working at it. You're going to be really fascinated, and all of a sudden the tide is going to go out. I mean that literally. It's like watching the tide go out. What you're going to start seeing is like, why is everything starting to flow sideways on my slide, right? And the reason is that you're getting evaporation at the edges, and that's creating capillary action, and it's pulling the fluid, and of course everything goes along for the ride, okay? Now it takes a few minutes for this to happen, but it's not like, oh, I prepared my slide, let's go have lunch, now I'm going to come back and look at it after lunch. No, you will not have a viable slide. We really want to keep our slide analysis within the first 20-minute window of and half an hour later, I can, no, it's like you're not going to be happy with the results, okay? Is all this making sense? Okay.
So,
Yeah.
the vial,
Yeah.
if we capped it, we can make a second or third slide?
Well, is that if you do that, it's not going to be the same because you've already agitated it once, you have to agitate it again. It's like you're comparing apples to oranges at that point in time. Your best thing of doing is go get us fresh and just do it from scratch, right? It's like stand back, we're doing science, right? So it means we want it to be repeatable and as controlled as possible. So if you just shake it back up, now you got a differently agitated sample than you did the first time, and so forth. So yeah, your best is to, that's why if you think you might need to take multiple samples, then get enough soil that you can actually start from, it doesn't take that long to do this.
Yeah.
So it's pretty simple, okay?
Is there a reason you would put drop 2, 3, and 4 on 3 different slides or something, or probably not?
If you had multiple students that all want to look at the same slide, yeah, sure, you could do that, right? Okay. Usually when you're doing it for yourself, it's like, no, you got one microscope and one set of eyes, and so you just create one thing, okay? So, and then it's like examining the sample, okay? When we're looking at a sample slide, you want to assess 5 basic things: the amount and quality of organic matter, the number and type of bacteria, the number and type of fungi, the number and type of protozoan, and the number type of nematodes, okay? What we typically do is we go to the 400x, and we say at 400x, the smallest bacteria you can see is about 1 micron in size. That sets your size, okay? Always remember to focus through the whole depth of a slide, because there's going to be a lot in one field that you're not going to see if you only focus on. Remember that, like, even though we think of it two-dimensional, because you got the slip cover, and it's like, oh, it's like, no, that water column at 400x between the slide and the slip cover is a multi-story building at 400x, right? Which means that to see everything, you might have to actually focus from the top to the bottom. And you're going to see different things coming in and out of focus as you do that. It's almost like you're looking down through a building, and as you focus, you're like, well, there's the 17th floor and the 16th floor, and it's like completely different things can be going on every floor, right? And so you have to focus down through there. You're like, well, that wasn't happening. I was like, it was down on the 2nd floor. You never focused down there, right? Yeah, there was a lot of that going on, but it was down on the 2nd floor, and you never got there. You only looked at the top half of the building, right? So that's why we focus through the whole thing, right? Remember to focus through. The water film from a single drop of water under cover slip is usually somewhere around 15 to 20 microns in depth. So assume it's like a 20-story building you're looking through, right? Which means you'll miss a great deal if you don't. So, and there's a whole thing here I'm not going to read to you about. You can, when you get into your groups, you can start looking at, right? How to go through all of this, how to look for nematodes. This page is your good guys versus bad guys, okay? I gave you a little table, and it says bacteria, good guys. Round and rod-shaped bacteria are most often good. Vibrio, that's the comma-shaped, are an indicator of anaerobic conditions. The spirillum, those are the spirals, can be dangerous disease-causing organisms, and spirochetes, another form of that snake-like one, are indications of compacted anaerobic soils. The fungi, we're looking for 3 microns or larger in diameter and colored. If it's less than 3 microns, 2.5 microns or less, or clear, then it's most likely anaerobic fungi. The protozoans we're looking for are flagellates and amoeba. We don't want to see ciliates. Those are anaerobics, right? Remember when we did a slide earlier, I said, are ciliates good or bad? They're on the bad guy list, right? And then nematodes. We want to see bacterial feeding nematodes, fungal feeding nematodes, and predatory nematodes. We don't want to see root feeding nematodes, right? Look for the knob at the base of the spear in the mouth, right? Okay, and then there's some other notes and everything else here I've given you. Every station has that, right? So what I want to do is we're going to quickly make certain we get our station set up back here, and we can get the. And what I need is I need the 2 containers of water back here. And
Yes.
are those them? Okay, let's go ahead and take them back there. Let me see what else I got here. All right. This is, by the way, notice that I keep all of my microscopy kit in a fishing tackle box because, I mean, I have all kinds of fishing tackle boxes just for keeping track of equipment. And so all the microscopy stuff goes in this fishing tackle box so that I don't lose it or I figure out where in the world it went. When I'm ready to go out the door, I'm like, well, there it is, right? So cheap fishing tackle boxes are a great way of keeping all this small little stuff together, right? Okay, so here are two more. So we got 4 of these. Okay, I've got 2 more pipettes. Let's take all these back and start getting the station set up. I've got slip covers, and these are our slides right here. See if there's anything else I need to get out of here. Nope, that's probably going to be good. Okay. Ah, okay.
Take these.
Yeah, so be real careful with those. Those are slip
I'll
covers.
be so careful.
Yeah, and then this is the lid for that. So just like that. There you go.
Yep.
And then, yeah, grab those. Yep. Okay. Nice.
We need a tray to rinse in.
Yeah, so you'll need a tray that you can like put the rinse water in. Okay. That'll work great. Yeah. You can just rinse water into that. Okay. Right, and we have samples. So let's go ahead and I want to see who's going to prepare our first sample slide. Huh?
Okay.
I will.
Okay. Right. Okay, so what you're going to do is get our first sample that we're gonna use, right? Okay.
Labeled? Does it need to be labeled?
I mean, I will always label them when I take them. We're gonna need little cups like this.
This is wood compost.
Okay. We're gonna need 2 cups. Actually, yeah, 1 for clean and 1 for rinse. Okay.
This is just, this is getting the sample on the slide.
Perfect. Okay. So what we're gonna do is we're gonna take our water and we're gonna put water in each cup. Okay, and then I like to put my clean water over here, out of the way, and put a pipette in it, and that's going to be my clean water pipette. Okay, this is going to be my rinse water, and I'm going to pipette in it, and these never cross. Okay, this is going to be our rinse pipe. This is going to be our sample pipette, and that's going to be our clean water pipette. Okay, so when we rinse something, we're going to rinse it in here. And then what we're going to do is we're going to come here and we're going to grab a slide. If I can get in here. Go ahead. Yep. Okay. And then hold it, and just put it on the top for the moment because we won't be ready for it for a moment. And then you're going to have to do this. This is super fitsy, but you're going to have to like pull this top pulls out of that, and we're going to get a single slip cover. They like to stick together.
Is this for holding?
That's actually you can actually write on okay. On that, like put a number on it. Yeah. It's here. I'll get it. It's fitsy.
I got a neck knife.
Okay. Yeah. Let me see if I can remember where the grip is. Oh, there it is. So I can get it in there. It's been a while since I've opened this.
I thought it was down on the bottom.
Yeah, that may be it. There it is. Okay, I got it. There it is. Okay. And now let's take that off. Yep. I'll let you go ahead and go with that.
And where do you want me to put this? Oh, this is wrapped.
Okay, let me see if I have another one that's already open. I thought I have a couple of those. Let me go see if I've got one that's already open.
Okay.
Usually what I'll do is I'll put it
There's 2
like...
slides there.
There's 2 slides in that one? Oh, yeah, there's 2 stuck together.
I can separate it now that I know.
Okay. It looks like 2 slides stuck together.
Oh, yeah, I see it.
Yep. There you
Looks
go.
like it's underneath his cover.
Can I set them both down like
Yeah,
this?
just set them down just like that. Okay, let me go. Yeah. Yay, good. Yes. Okay, let's just pull this over here where we can open it all the way up.
Yeah. I was going to ask you earlier if you had a USB cable.
Let's see if that one's open. Oh, I tell you what, you can just go ahead and use these.
Okay. Yeah, that's still wrapped too.
Okay.
I usually have stuff like that.
Yeah. Perfect. Thank you.
Tiny fingers for that.
As it might not be contaminated.
Usually we'll use, let me see if I have one of them. I have, yeah, I usually have one of those. Let me see if I've got something here we can use. Let's see. Yeah, I don't. Those didn't get in here because I have those out in my lab.
What should I be looking for?
Basically, here, we'll just do it like this. Yeah, just take and open that up and shake it in.
Shake it up, baby. Shake it.
Yep. We want to get about 2. Well, yeah, we might be able to get. Yeah, it's so small, right?
Compaction.
Mm-hmm.
Can you use something to spoon it into there?
Use my knife.
Yeah,
Let's
That's
say.
why you need a knife.
Let's say I was about to pull this one out. But yeah.
You
You
want to hold?
I'm gonna
I can
put it-
hold it.
Usually, I have these little wooden pallet. Yeah, tamps. Yeah.
Maybe
Yes.
one of these body butter people has a little wooden spoon.
Grab the compost one that's gonna be.
Kind of hairy like this, right
Yeah.
off the top.
Mm-hmm. Okay, if you want to be able to prepare a slide, come watch because we're going through it right now.
Yeah.
Yeah.
I might have to shake some out.
Yeah. So
So do you want 2 compact?
yeah, two compact is what we really want.
Okay.
Yeah.
Here, excuse me. Yeah, we're got way more too much.
Mm-hmm.
Oh. That little turd ball is probably it right there.
Mm-hmm.
That's still too much. Yeah, it's 2's way up there. It's not enough.
Yeah, and a little bit more. A little bit more.
I'm gonna put all back in what I took out. No. Get a little aluminium in my sample.
Mm-hmm.
It's fine. Everything's fine. Yeah.
Happy with it?
It fits in so many occasions.
We could use a tiny bit more than that. 'Cause, yeah, just a little bit more.
Don't you have glasses on?
It's going to end up being the same amount you had from the get-go.
That's what I said. Yep.
Okay, we'll just roll with that.
Yeah, we got.
Okay.
A little bit.
Now what we'll do is we'll take our clean water.
Always close your sample right after you're done with it.
Yep.
Thank you.
We'll take our clean water and we'll just, we'll come in here. We'll get some water. We'll bring it up so we can look at it at eye level, and we'll just start adding our water.
All the way to 10.
All the way to 10.
So does that make it a 1-to-4 ratio or?
Yeah.
Down to drops. Coming in hot.
Yep. Okay. There we go. Okay, then we put our clean water away. Get our cap. Put our cap on there. And now you do your agitation.
Stand back.
all the way out, just like that. One, yeah, exactly. A little
With
faster.
intention.
Yeah, little faster. There you go. That's about right. And do about 30 times.
Where's your pocket centrifuge, Patrick?
Trying to count. 18, 19, 20.
21, 22, 23, 24, 25, 6, 7, 28, 29, 30.
Good. And now just hold it. What we're going to do is, I'll hold up here. Well, you notice it's like,
Now look what you've done.
yeah. Now, if we wait a few seconds, we're going to start seeing that some of that sand and the heavier stuff starts to come to the bottom, right? And I'm just going to let it do that for just a little bit because when I take a sample, what I want is more of the suspended, of the liquid without like big sand stuff. Right.
On the top.
What we were trying to do with the agitation, that's going to be, yeah, we're not going to sample that. What we're trying to do with that agitation is to get a lot of the life off of the surface of a lot of those and up into the water so we can get it onto the slide, right? And by trying to be repeatable on agitation, we're trying to get something of a similar percentage every time, right? That's what I'm saying. If you were really doing this in the lab, you'd be using lab equipment to get it much more precise. But you notice, you see it's starting, you see the dark at the bottom and so forth. Okay, now I'm to the point where I can sample that fluid up here. I don't want to get my pipette down in there. It's just going to get bogged down. So I'm going to sample from right up in here, okay?
So the aggregates are on the bottom, and you've still got some organics floating, but they're definitely not sand and silt.
Correct. That's what we're trying to do. So take this off, and now we're going to use our sample pipette right here, right? Okay, and I've taken it and I've rinsed it out really good. In the ideal world, we would have a different pipette, or we would clean the pipettes between use, right? So I hate the fact that these are supposed to be single-use pipettes. And in the lab, they are. You can get a box. So I have a box of 1,000 of them, right? And they're supposed to go in there. I hate that. So when we did it for Wheaton Labs was like, no, throw away plastic. And so we got them washable pipettes, and you have to wash them between every use. Okay.
Did you autoclave them as well, or just wash them?
We just wash them, yeah. Because we're not doing surgical work. We're just doing basic lab work. So what we're going to do, this is the fitsy part. I'm going to come in here to about here, and I'm going to go, I'm going to need the slide here in just a second. There's 1, 2, 3, and now on my 4th try, I'm going to come up, and then I'm going to get 1 drop off there, and then I'm going to do 1 drop there. And I'm going to do it pretty vertically because I don't want to do, I want it to a vertical drop like that. Now I'm going to take, and if you've done this, yeah, go ahead and you're going to take about there. It should, there you go. You see it. Now just drop it. Boom. There we go.
It's like a pro.
Yes.
Yeah, I got it.
All right. All right. There we go. Thank you. And then we'll let you take that. Okay. Now what you're going to see is it wasn't quite perfect, but it got spread out over most of the slip cover. Okay. As you get a little cleaner with it, you'll get it like to go out perfect. But that's good enough for us to go throw on the microscope. So let me go throw this in here and let's just kind of start seeing what we're seeing.
Probably all sorts of little Rormans in there now. Little wigglers.
You ever use a microscope to look at your knives?
I have. I've got a digital microscope that I use.
The good guys go to the right. Bad guys go to the left.
There's nothing there.
Some of them say, why can't we just all get along?
Ooh. Yeah, I'm not sure which particular one is.
Okay. Let me see here. There we go.
You're starting with which power?
I'm at 400 right now. I'm going to pull it back and see. Okay, let me look. Why are you doing that?
You
It's
need to
like,
look with your normalize just to get the
yeah.
sample underneath.
I was trying to figure out. I'm gonna go to you here. There we go. Let's see if I can do that. There we go. Why are you misbehaving? Okay.
There they are.
Yeah. Okay.
There's some stuff.
Oh my word.
Buddies. Yo,
Hey, that was new.
Oh, what is it?
Hey.
Whoa.
it's DNA.
Look like it, but yeah.
I know, but it's fun to imagine.
Yeah, it's what exactly what I thought it would look like.
It's like cloud gazing.
I found a dragon. Yeah.
Oh, there's somebody.
Yep.
Fungi.
Yep.
It's got color. It's the good guy.
Yep. Okay, so the first thing you're going to notice is that the bacteria are kind of vibrating. Can you kind of see that?
Yeah.
Oh, yeah.
Okay, so you see a lot of little coccybacteria, right?
Yeah.
Okay, we want to see the coccybacteria. Those are good guys. We're seeing little bits of silicates and microclay particles. We're seeing little tiny bits of soil organic matter. That little clump in the upper middle that's kind of that golden brown color is soil organic matter.
Are the bacteria vibrating because they're thinking?
Yeah, they're actually moving. Yeah.
God,
Okay.
so creepy.
Right. Okay.
Oh, look, there's a
Yep.
little thing that's like,
Goodbye.
What's that thing rotating towards the middle?
Which one? Right. Oh,
It's on the left.
yeah. Yeah.
Now.
That's a larger rod
Okay.
bacteria. It looks like it's not big enough to be a protozoan.
the big long one.
Do you want a laser pointer?
That's a fungi.
Let me see what we got.
Maybe it's hair.
Because it's colorful.
Yeah. Although the long thin thing is a fungal. Yeah. It is over 3 months thick, and so I'm liking that. That's looking good.
Did you that blob that the fungi's pointing into focus that keeps moving? Oh, it just turned white. Whoa. Is that the tide?
That's the tide. What's happening is since we didn't get it perfectly even, capillary action still trying to pull, right? Trying to pull the sample around, and if you look at it, it's pulling towards. Yeah.
Wow.
So what I'm doing is I'm using the fine focus to kind of like just go through the pile, and you'll see as I go through, different things start to come in and out of focus, right? Yeah. Right. Here, let me see if I go. Whoop. Oh, this is being really finicky. There we go.
There's a lot of oval things.
Yeah.
There's a large something or other on the edge of the slide.
Yeah. So you've got some microsilicates there. Those the sharp-looking things. Those can be clays and microsilicates. The things that look a little bunchy and aggregated are soil organic matter. I'm seeing a lot of bacteria. I'm seeing a little less fungi than I would expect for wood chips. Where did this come from? And wood compost. Okay. Let me just scan around just a little bit more, see if I can see. Okay.
Oh, there's a big guy. Whoa.
Yeah. Okay. So that looks like a cyst of some sort. Okay. That can be a spore or a cyst, so it can be something that's or of a protozoan.
Alan,
Yes.
the compost pile that came from is
Yep.
only about 2 months old.
Okay.
If you want to do our compost from last year, I can show you.
Let me
That
go.
was a sample that came from a compost pile in the woods.
There's a good example of soil organic matter right there. Let me focus in on it where you can see it. But yeah, you can see it's three-dimensional. So if I focus through it, you're seeing that's soil organic matter, which is nice. Okay. You can. Yeah, you'll.
This from the woods.
Yes.
Yeah.
2 months old.
Yeah. So you will find that yes, this is sort of like the soil organic matter clusters are like the coral reef, right? Everybody likes to hang out around the coral reef because it's a place that supports life. So don't be surprised when you see a lot of like bacterial activity right around it and so forth. That's pretty common, and you might see other kinds of activity of protists and things like that pretty close into the soil organic matter. Okay. So
doesn't necessarily mean they're feeding on that.
No, not necessarily. They could be, but it also it's just it's a good environment for them to be. You know, it's a good place. There's a lot of reasons that soil organic matter is. It's kind of like good housing, a good structure for them, and so forth. And yeah, so you'll see a little more activity right around it. Okay. Now, today we're not going to have enough time to go through all the identification of everything because you can see there's hundreds of things to see here, right? And if we want to do a full class on this at some point in time, which is likely to be a multi-day class, then what I would do is bring another instructor from our institute, Chase, who is certified as a soil microscopy technician. He can write certified lab reports on this. Okay, so he's the one who's had like ridiculous amounts of microscope time, and will in terms of identification. Like, yeah, he's really the expert. I defer to him because he's had so much training in that now. But this what you're starting to see is, yep, we're getting some soil organic matter. Yeah, we're getting a lot of bacteria. No, I haven't seen anything massively anaerobic. That's good.
Is the nematodes on the left moving under its own power, or is that drift from
The way you look at that is like almost defocus. And if you look at it broadly and there's like a bunch of stuff all moving together in the same, then that's drift. Right? If you've got everything just kind of vibrating, that's certain. If you have things moving like different directions, then they're moving under their own power, right? After a little while, you'll get to the whole point of like, yeah, it's drift because you'll see like the whole thing going like this together, right?
that little dude went inside?
Yeah. Okay. So somebody asked about nematodes.
Yeah.
We are currently at 600x. This is way too far in to see the nematodes easily, because they're really big compared to everybody else. So what we would normally do is we would go back out to something more like 200x and scan for nematodes. Okay, you'll see that on your instructions. What that would look like would be to take. Okay, actually I've got the 100x. Okay, I could come here to 100x. Okay, now we're looking at the same scene, and right in the smack middle is that organic matter we were looking at. And now we can see a lot more really quickly. I can kind of just start moving around, and we can see broad things. Oh, there you go. Okay. That looks like right there cellulitic material. That I'm moving towards the center. See the structure of it? Has this like looks like a little chain of yeah. That looks like leaf matter or woody materials that's still being broken down. Okay, right. But I would usually just scan the whole slide this big to see if I'm seeing any like nematodes, because nematodes will stand out to you because normally they're like snakes, like a snake that's excited, like an excited snake on a slippery surface. That's the best way of it. They're like doing this, okay? They're not going that far, right? Okay. So you're seeing here some fungal hyphae. You see that? Yeah, actually, yeah, mine was like not bright enough to show up here, but like
probably won't show on the screen.
Yeah, but say on the screen. I'll just point with my finger.
Okay.
Thanks, Jack.
You're welcome for giving me the screen. You can't point to.
Like we have to go in on that. That's it's kind of rough. I need to go in and look a little closer. I want to look at things like this to see that's uneven. Even from here, I'm seeing it's uneven. If it's uneven, then it's probably not fungal hyphae. It's probably something like leaf material or some other woody material that's been shredded down. When you see good fungal hyphae and you go in at like 400x, you're going to see that it looks almost like a piece of bamboo. It's just like this. It's really, even. It's got the segments and so forth. That's really what you're going to see, okay? Big chunks of, like, this is a bunch of soil aggregate. You got soil organic matter, and you might have some stuff still being broken down in there. But that's soil organic matter. So I'm seeing a pretty good amount of soil organic matter. I'm seeing a lot of bacteria and so forth. I haven't seen nematodes yet. We haven't seen a lot of protists yet. They are going to be medium sized. They're not going to be as big as a nematode, but they're going to be a lot bigger than our bacteria. Okay.
For nematodes wiggling, that'd be exciting.
Yeah, should
Yep.
we still be seeing things moving at this?
This is that 2-month-old compost, though.
Yeah,
Yeah.
I have the year-old stuff back there
Yeah.
that you might find more.
Mm-hmm.
Like a forest soil sample. I mean, this is.
slide made?
Yeah, go ahead and let's. Oh, there we go. Okay, now we got somebody. Now we got somebody. Yeah.
Oh my God, Godzilla's got nothing on him.
Oh, hold on. Wait a minute. Hold on. Let me go in a little closer on him. Let me see if I think that. See that? I think it is.
It's
It's got
cute.
paws or something.
I think the Planet Killer
Caterpillar.
series was based on a nematode.
You may think this is funny, but this one's deadly.
He's stuck on that.
That
Like, get this off of my.
may be a tardigrade. Hold on.
Oh, all right. Maybe a who?
Tardigrade.
I see. That's. Yeah.
Oh, dude, get me out of here.
Yep.
Stop. I'm so stuck.
So like next year we're just gonna do a whole session without the intro where we just do live watching of soil
We need some
microbiology
creepy-ass
and
music
just
to go with
record the
that.
noise. Whoa!
This is one of those bear things
So-
in space.
The way I had done microscopy for a good while,
Yeah.
but my real introduction to soil microscopy was well back before Elaine Ingham started the Soil Food Web School. We got to go spend 3 days with her just doing this.
Yeah.
Just sitting here going through all this stuff. Yes, that's a tardigrade.
He broke away.
Okay.
Is that a white fungi over to the left of it? That's the bad guy?
Let me take a look there. That
You're looking to the right, maybe?
yeah, it's like I won't have to try to
To the left on the screen.
yeah. Yeah, that is a bad fungal. Like you can see the segments and you can see the inside of it,
Yeah.
so forth. But yeah, you're lucky. You guys are really lucky. You got to see a tardigrade live.
Moving a little bit.
Yeah, they're called water bears, right?
Yeah.
Yeah.
That is a starter.
Yeah. Okay. So anyway, this is what you're going to be doing. You're going to start, and you're going to be doing most of your work at 400x. This is where we are right now. We're at 400x, and we'll back out to 100x to scan for big features like nematodes and so forth. Okay.
Would you see something like parasites in water? Like what magnification would like parasites that you?
According to what the parasite is.
Yeah.
There's some parasites that are a lot bigger than others and so forth. One of the things you can do with your animals, and it's a whole different class, is fecal microscopy, where what you do is you take their fecal matter and you prepare it in a particular way and put it on a slide, and you just look for the parasites, and you can visually do parasite counts from the fecal matter, okay? And you can actually measure, so like if you're doing goats or sheep or whatever, and you've got the microscope, then one of the things to be doing is there are kits that let you collect the fecal matter, bring it in, and then put it onto a slide in such a way that you can actually count the parasite load, and you can keep track of the parasite load in your livestock. Okay, so it's a very, and it's very handy. You can use the FAMASHA eye charts for eye color. That's also very handy. But this is a really great way to see what the particular parasite is, because you can literally see it.
1-year-old
Yes.
compost.
1-year-old compost. Okay, let's see what we got. I'm gonna hand you this so you
This
can go.
is Jack's bioreactor build compost from last
Okay,
year.
I'm gonna
Don't
take.
be bad. Don't
You'll
be
take
bad.
that.
Hey, I just didn't take care of it. All I do is
Save the tardigrade.
Oh,
you can go to space and back, man.
I don't know anything about tardigrades.
Okay.
Oh crap!
Tide.
Yeah, so you can see the tide going. It's like it's still you know, we put it on. It's now it's like what was happening was that capillary action was spreading everything out, and now it's kind of like so we caught it just in time. Okay, so we're seeing immediately a pretty good amount of like little clusters of soil organic matter.
Are you still at 400?
Yeah, no, I'm back at 100 right now. When I pull a slide out and put another one in, I typically take it back to 100 so I don't bump the objective. I don't want to like slide, you know, like bump the slide into the objective and scratch the optics. So I push it back to 100 so I can get the slide in and out without any danger. Right? Yeah. Okay, let me do a little bit here. So yeah, I'm seeing a lot of good soil organic matter, little aggregates all over the place. Let's just do a quick scan around and see what we're seeing. Okay. You know, some of this is looking like just organic material that's still being shredded down.
At that objective, do you know like how many fields would be on that size slide? Like, are you talking 200 or 1,000?
Well, I'd have to do the math to figure out exactly. We typically, if you're doing a lab report, you typically are going to sample like 20 fields. That's pretty typical. And you work your way across the slide doing that. I'm just scanning around so we can see interesting things. So there are some, looks like there's some fungal hyphae. They seem to be clear. That's interesting. Just going to scan across real quick. See if I can see any other big features that are interesting. And we'll go in tight so we can, let's just go a little more tight on that particular bit of aggregate and see what it's going to give us. Let's go into 400. And then give me a second to, there we go. Okay, now I got to focus here. There we go. All right. Okay, so you're seeing, now you can see bacteria in the back just kind of like this. Okay. You can see soil organic matter popping back and forth. So, and we were seeing some different fungal hyphae. I was seeing some bad and some good both. Whoop. There it is. Okay. There you go. Soil organic matter, nice. Okay. We go through, you can, see that little thread down there?
Yep.
Okay, that's a thin one. The question when you see really thin and you see really thin and you have to ask yourself, can you see the inside and is it segmented? If it's clear looking and you can't see the insides and it's very segmented, not segmented, it may be actinobacteria instead of fungal hyphae. Yeah. Okay. Everybody getting the idea of what we're doing here?
Yeah.
Okay, what I'd like to do is let everybody go play with microscopes then. Because I want you to get hands on microscope for a second. So let's go ahead and break up. We got 3 microscopes up there and this microscope here. I'll stick with this microscope. We have 3 people upstairs. How much time we got? Or are we done?
Negative.
Already done.
20 minutes.
This is so cool.
Here's
Yeah.
my thought.
Yeah.
Because Oakland and I are the last 2 sessions of the day.
Yeah.
Okay. Yeah. If we could start concentrating more upstairs,
Mm-hmm.
and maybe 15 minutes, she can start fibers down here or whatever. That gives us time to look at the microscopes. One of those microscopes I don't think is put together right.
Okay.
So we should have you look at that
Yes.
station.
And what we can do is this. I can leave out stuff for right now.
Yeah.
And then later on today, we can come back and pull the microscopes out and start working with it. It's like normally when I've taught this, it's been like, yes, and today is soil microscopy, right?
Yeah.
So, but I mean, I wanted you to at least get the thing of saying, oh, this is doable, right? There's nothing here. You don't have to have a PhD in biology to do this. It can be fun. It can be great fun to do with the kids, right? And so forth. I need to get focused back in where you can see because I was.
By the way, we figured something out. The minute it's all done, if you want to point to something on the screen, use your cursor.
Yeah.
Oh yeah.
You got a finger pointer thing.
Yeah.
Is there any amoeba?
I haven't seen an amoeba yet, but you will get them. Nothing that I have seen so far has been in, like what I would say is like, wow, that's what I want to see.
Yeah.
Right. I would want to see. I mean, what we've done before is we've gotten like some super high end compost from, either something we've done or we get it in from something like Catalyst Bioamendment. We put it on there and we show that to you, and it's like, oh my god, and then everybody gets to ooh and ah, right? So we've done that before at a thing. It's like, yeah, we ordered, a thing of Catalyst Bioamendment compost, and then we bring it in, and then we're like, how about this? And everybody's like, what's that? What's this? It's like that's really fun to show to a group because now you've got like protozoa, the proper protozoans. You got lots of fungal hyphae, and you got nematodes, and you got just all the stuff, and it's like the circus, and you get to everybody gets really excited.
For hobbyists who want to do this, is there a good guide to what you're looking at there?
So a couple of things. One is what I put this in the Telegram group for you, so you'll have this, and then there's actually now a lot of YouTube channels where
there had to be a YouTube university.
there's a lot of YouTube channels where people are showing off what they're seeing and they're showing identification and so forth. That's a great way of doing it. There's a couple different. If you really want to get serious about it, Soul Food Web School has a full certification course. Matt Powers has a course in regenerative soil microscopy. He's using a much higher end microscope with epifluorescence and doing some more advanced stuff. And that's for if you want to really get serious and spend like $4,000 to $5,000 on a microscope. But he's doing some stuff that we can't do here because he has epifluorescence and so forth. Okay, so what I'm going to do is I think let everybody move upstairs so that you guys can get set up. I'm going to take this one down so it gets out of the way. We'll just use the microscopes upstairs for right now, okay?
Is it in your way? I was hoping to use the table. Okay.
Yeah. So it'll just take me a couple minutes to get this one put away. So I'm going to take this slide.
Slides down here first.
Yeah.
Before we go upstairs.
Who
Yeah.
did Michael Vertrees learn from?
Yeah. All right. So I'm going to give you this slide right here. Thank you. Yes, please.
Pretty close, yeah.
Yeah, and these are new slides I haven't used, so you just Oh, that's the old one then. Okay, hold on.
No, I've already watched the old one. That's the one you were demoing with dust.
Okay, so I'll put that right there. I'll just keep this one dusty.
Okay.
And I'll put it right here. All right. So yeah, head upstairs and start playing with those up there, and we'll start creating some slides, and just give yourself a few minutes to like figure out how to make a slide while Opalyn gets set up for her session, and that'll let me get this out of her way. Okay.